Dear students, myself Anuj Modi, Assistant Professor, Adina Institute of Pharmaceutical Science, Sagar, MP, with an interesting and learning episode of B.Pharm on the important topic of herbal drug technology, which is about chemistry, biogenesis and pharmacological activities of medicinally important monoterpenoids, sesquiterpenoids, diterpenes and triterpenoids, and which is one of the most important unit of section Pharmacognosy B.Pharm 8th semester. This is prepared by me and Dr. Vimal Kumar, Professor and Head, Department of Phytopharmaceuticals and Natural Product, Institute of Pharmacy, Nirma University, Ahmedabad, Gujarat, India. Let's start our episode while taking a look at what we are going to learn today. Module 1, Terpenoids, Introduction and Classification. Module 2, Biosynthesis of terpenoids, monoterpene, sesquiterpenoids, diterpenes and triterpenes. Module 3, Chemistry, Biogenesis and Pharmacological Activity of monoterpenoids. Module 4, Chemistry, Biogenesis and Pharmacological Activity of sesquiterpenoids. Module 5, Chemistry, Biogenesis and Pharmacological Activity of diterpenoids. Module 6, Chemistry, Biogenesis and Pharmacological Activity of triterpenoids. Let's start with the module 1, Terpenoids, Introduction and Classification. Terpenoids are volatile substances which give plant and flower their fragrances. The term terpenes was given to the compounds isolated from turpentine, a volatile liquid isolated from the pine tree. Terpenoids are the hydrocarbon of plant origin, which is simply represented by general formula C5H8N, as well as their oxygenated, hydrogenated and dehydrogenated derivative. On the basis of value of N or number of carbon atom, terpenoids are classified as monoterpenoid, sesquiterpenoid, diterpenoid, sesquiterpenoid, triterpenoid, tetraterpenoid and polyterpenoid. Monoterpenoid contain total number of 10 carbon atom and 2 units of isoprene unit, while sesquiterpenoid contain 15 carbon atom and 3 unit of isoprene. Diterpenoid contain 20 carbon atom and 4 unit of isoprene. Sesquiterpenoids contain 25 carbon atom and 5 units of isoprene unit. Triterpenoid contain 30 carbon atom and 6 unit of isoprene, while tetraterpenoid contain total 40 carbon atom and 8 unit of isoprene unit, while polyterpenoid contain more than 40 carbon atom, which is represented by C5H8N. Each class can be further subdivided into subclasses according to the number of rings present in the structure. Those are acyclic terpenoids, they contain open structure; monocyclic terpenoids, they contain 1 ring in the structure; bicyclic terpenoids, they contain 2 rings in the structure; tricyclic terpenoids, they contain 3 rings in the structure; tetracyclic terpenoids, they contain 4 rings in the structure. Some of the medicinally important monoterpenes, diterpene, triterpenoids and tetraterpenoids are given below. Monoterpenoids such as acyclic monoterpenoids, myrcene, citral and geraniol. Example of monocyclic monoterpenoids are limonene, menthol, alpha-terpenol, while example of bicyclic monoterpenoids are alpha-pinene, camphor. Example of sesquiterpenoids, acyclic sesquiterpenoids such as farnesyl, monocyclic sesquiterpenoids, gingiberine, bicyclic sesquiterpenoids, cadhenin, tricyclic sesquiterpenoids, artemisinin. Now, the example of diterpenoids such as acyclic sesquiterpenoids, phytol, monocyclic sesquiterpenoids, vitamin A. Example of triterpenoids such as quercetin, cucurbitrin B, azadirachtin. The simpler mono and sesquiterpene is the chief constituent of essential oil obtained from the sap and tissue of certain plant and trees. The di and triterpenoids are non-steam volatile. They are obtained from the plant and tree gums and resin, while tetraterpenoids forms a separate group of compound called carotenoids. Now, let's move toward our next module. Module 2, Biosynthesis of terpenoids, monoterpenes, diterpene, triterpene and tetraterpenes. Volatile oil are nothing but the compounds which evaporate at the room temperature, so they are called as volatile oil. They are responsible for characteristic odor of the drug, so are called as ethereal oil. Chemically, they are terpenes made up of hydrocarbon, that is isoprene unit represented by C5H8, and their oxygenated and methyl substituted derivatives. It has been proposed to call both the terpenes hydrocarbon and their derivative collectively as terpenoids. Biosynthesis of volatile oil arises from acetate moiety, which is obtained from TCA cycle. On glycolysis, sugar molecule yields pyruvates, which enters into the TCA cycle and produces acetate moiety, which gets converted into acetoacetyl coenzyme A by thiolase enzyme. By using HMG coenzyme A synthetase, this acetoacetyl coenzyme A gets converted into beta-hydroxy beta-methyl glutarallyl coenzyme A. Further, this beta-hydroxy beta-methyl glutarallyl coenzyme A utilizes NADPH2 system and HMG coenzyme A reductase. After number of steps, finally gets converted into mevalonic acid. This is the rate limiting step of in this biosynthesis terpenoids, which is blocked by statins. Further, this mevalonic acid undergoes phosphorylation at 5th and 3rd position and gets converted into 5-phospho-mevalonic acid and 5-phospho-3-phospho-mevalonic acid. During this process, ATP acts as a phosphate group donor and reaction is controlled by mevalonate kinase and pyrophosphomevalonate kinase respectively. On decarboxylation by decarboxylase enzyme, 5-phospho-3-phospho-mevalonic acid gets converted into isopentyl pyrophosphate. By isopentyl pyrophosphate isomerase, this isopentyl pyrophosphate gets converted into 3,3-dimethyl allyl pyrophosphate and act as a precursor for the biosynthesis of hemiterpenes. When 2, 3, 4 and 6 molecules of isopentyl pyrophosphates are combined together, leads to the biosynthesis of geranyl pyrophosphate, farnesyl pyrophosphate, diterpene and sequelin, which act as a direct precursor for the synthesis of monoterpenes, sesquiterpenes, diterpenes and triterpene type of volatile oils. The sesquiterpenes, triterpenes and polyterpenes appear to be produced in the cytosolic and endoplasmic reticulum compartment, whereas isoprene, the monoterpenes, diterpenes and certain prenylated quinones originated largely, if not exclusively, in the plastids. Dear friends, up till now you have gained a complete idea about different types of the terpenoids and their biosynthesis. If you have missed any of the module or content, you may log on to our website www.cec.nic.in to download videos, FAQs, LOR and other content so that you can remain updated with this chapter. Now let's move ahead towards our next module. Module 3, Chemistry, Biogenesis and Pharmacological Activity of Monoterpenoid. It has been found that a large number of monoterpenoid derivatives belonging to these categories invariably occur in naturally occurring plants in the purest optically active form. However, certain plant species do have both enantiomers, such as Pinus species contain dextro and levorotatory pinene. We will discuss here in detail about menthol. Menthol, synonym L-menthol. Biological source, it is found in the peppermint oil obtained from the fresh flowering tops of the plants commonly known as Mentha piperita or other allied species of Mentha belonging to family Labiatae. Now, the chemistry of menthol. Color, colorless. Odor, pleasant peppermint-like odor. Taste, characteristic, aromatic and cooling taste. Shape, it occurs as hexagonal crystal, usually needle-like prism, crystalline powder and fused masses. Menthol has 3 chiral centers, hence it would give rise to 8, that is 2 to the power 3 optically active isomers and 4 racemic forms. Those are levo and dextrorotatory menthol, levo and dextrorotatory isomenthol, levo and dextrorotatory neomenthol, levo and dextrorotatory neoisomenthol. Menthol on oxidation gives menthone, a ketonic derivative. Therefore, the resulting menthone must exist into 4 optically active isomers and 2 racemic forms, and all these have been actually prepared. On dehydrogenation, menthol first on dehydrogenation yields 2 isomeric form of paramenthine, which on subsequent dehydrogenation gives rise to paracymene. Reduction, on reduction with hydroiodic acid, it yields paramenthane. Now, we move towards the biogenesis of menthol. Biosynthesis of menthol was investigated in Mentha piperita. It begins with the synthesis of terpenes limonene, followed by hydroxylation, and then several reduction and isomerization steps takes place. Limonene synthetase catalyzes the cyclization of geranyl diphosphate to levorotatory limonene. Limonene 3-hydroxylase using oxygen and NADPH then catalyzes the allylic hydroxylation of limonene at the 3rd position to synthesize trans-isopiperidinol. This levorotatory trans-isopiperidinol dehydrogenase further oxidizes to hydroxyl group on the 3rd position using NAD+ to make levorotatory isopiperidone. This levorotatory isopiperidone reductase then reduces the double bond between carbon number 1 and carbon number 2 using NADPH to form dextrorotatory cis-isopulegone. This dextrorotatory cis-isopulegone isomerase then isomerizes the remaining double bond to form dextrorotatory pulegone. Pulegone reductase then reduces this double bond using NADPH to form levorotatory menthol. Menthone reductase then reduces the carbonyl group using NADPH to form menthol. Now, we will study about various pharmacological activities of menthol. Menthol ability to chemically trigger the cold sensitive TRPM8 receptor in the skin, which is responsible for the well-known cooling sensation. It provokes cooling effect when inhaled, eaten or applied to the skin. Menthol also produces analgesic effect mediated through a selective activation of kappa opioid receptor. Some studies shows that menthol acts as a GABA A receptor positive allosteric modulator and increases GABAergic transmission in PAG neuron, which in turn decreases the pain sensation. Menthol also blocks voltage sensitive sodium channel, reducing neural activity that may stimulate muscles. Menthol also shares anesthetic properties similar to propofol, which act as a modulating same site of GABA A receptor. Menthol also shares anesthetic properties similar to propofol, which act by modulating same sites of GABA A receptor. Menthol is widely used in dental care as a topical antibacterial agent, effective against several types of streptococci and lactobacilli infection. It is also used in variety of formulation for the variety of purpose, such as in non-prescriptional product for short-term relief of minor sore throat and minor mouth or throat irritation, for example, lip balms and cough medicines, as an antipruritic to reduce itching, as an antispasmodic and smooth muscle relaxant in upper gastrointestinal endoscopy, in medication used to treat sunburn as it provides a cooling sensation, as a smoking tobacco addictive in some cigarettes brands for flavor and to reduce throat and sinus irritation caused by smoking. Menthol also increases nicotine receptor density, increasing the addictive potential of tobacco products, in certain medication used to treat sunburn as it provides a cooling sensation. Now move towards our next module, Module 4, Chemistry, Biogenesis and Pharmacological Activity of Susky terpenoids. In this module, we will study in detail about artemisinin. Artemisinin, biological source, it is obtained from the leaves and the closed unexpanded flower heads of Artemisia annua belonging to family Astraceae. Chemistry of artemisinin, molecular formula C15H22O5, molecular weight 282.33 gram per mole, melting point 150 to 157 degree centigrade. It consists of tetracyclic structure with trioxane ring and a lactone ring. 1,2,3 trioxane ring has greater potency than artemisinin, but it lacks stability in vivo. Unlike other antimalarial drugs, it does not contain heterocyclic ring with nitrogen atom. Chemically, artemisinin is a suscyterpene lactone containing an unusual peroxide bridge. This peroxide is believed to be responsible for the drug's mechanism of action. On reduction of lactone carbonyl group of artemisinin by sodium bromohydride into lactal group leads to the synthesis of dihydroartemisinin, which is a product. It could be either oil soluble or vital soluble. Introduction of methyl group at third number carbon of artemisinin increases its stability, but simultaneously decreases its bonding strength with heme, ultimately decrease in its antimalarial activity. Substitution at carbon number 9, alpha substitution analog of artemisinin showed improved antimalarial activity along with anti-leishmanic activity against Leishmania donvani parasite. 10-deoxy derivative of artemisinin showed better antimalarial activity, while 1-deoxy derivative do not show anti-leishmanic activity. Because the physical properties of artemisinin itself, such as poor viability, its hydrolytic instability, limits its effectiveness. So, semi-synthetic derivative of artemisinin has been developed, such as ether derivative and ester derivative. Dihydroartemisinin converted into ether derivative like artemether and artether. Both of these drugs are oil soluble in nature, which is used for oral, rectal, intramuscular administration. It cannot be used intravenously. While ester derivative, succinate ester of dihydroartemisinin, such as artesunate, was prepared to overcome the problem which is associated with the artether, that is, to develop water soluble derivative which can be injected intravenously. But drawback of this compound is that it contains unstable hemiester linkage and its rapid hydrolysis to dihydroartemisinin. To overcome these both problems, a para-carboxy-benzyl ether derivative of artemisinin, that is artelinic acid, was prepared, which is a water soluble in nature and does not show hydrolytic instability. Now, we will study about biogenesis of artemisinin. The biosynthesis of artemisinin is believed to involve the mevalonate pathway and cyclization of farnesyl diphosphate. It is not clear whether the non-mevalonate pathway can also contribute 5-carbon precursor, that is isopentyl pyrophosphate or dimethyl allyl pyrophosphate, as occurs in the other sesquiterpene biosynthetic system. The route for artemisinic alcohol to artemisinin remain controversial and they differ mainly in which the reduction steps takes place. Both routes suggested that dihydroartemisinic acid as the final precursor to artemisinin. Dihydroartemisic acid then undergoes photooxidation to produce dihydroartemisinic acid, then undergoes photooxidation to produce dihydroartemisinic acid hydroperoxide. Ring expansion by the cleavage of hydroperoxide and a second oxygen mediated hydroperoxidation finish the biosynthesis of artemisinin. Now, we move towards pharmacological activities of artemisinin. Mainly, it is the antimalarial drug. Apart from that, it is also used in the treatment of cancer. First, we will study about antimalarial effect. Artemisinin and its derivative are used as fast-acting blood seisonicides in the control and management of malaria fever caused by Plasmodium vivax strain. It contains unusual endoperoxide group and this react with the iron in heme, that is free form of heme due to hemolysis in case of malaria, giving rise to highly reactive free radicals. Parasite death is believed to the result from the reaction of these free radicals with parasite molecule such as protein and nucleic acid. Artemisinin does not react with the iron in hemoglobin, that is heme is bound with the globin protein so that uninfected red cells are unaffected. Artemisinin may however be embryotoxic so that the use of these drugs is not recommended in early pregnancy. These drugs are found to be active against both chloroquine resistant and chloroquine sensitive strains of Plasmodium falciparum. These drugs are found to show extremely encouraging therapeutic effect, especially in the treatment of cerebral malaria, by the virtue of their significant rapid clearance of the privileged parasites when compared to either chloroquine or quinine. Artemisinin can be used alone, but this leads to a high rate of recrudescence, that is return of parasites, and other drugs are required to clear the body of all parasites and prevent reoccurrence. The WHO has recommended artemisinin combination therapy, that is ACT, be the first line therapy for Plasmodium falciparum malaria worldwide. Several fixed dose artemisinin combination therapies are now available containing an artemisinin component and a partner drug which has a long half-life, such as artemisinin and mefloquine, artemisinin and lumefantrine, artemisinin plus amiodarone, artemisinin and piperacin. Now, we will study about the anti-cancer effect. Artemisinin has anti-cancer effect in experimental model of hepatocellular carcinoma. Artemisinin has a peroxide lactone group in its structure, and it is thought that when the peroxides comes in contact with high iron concentration common in cancerous cells. The molecule becomes unstable and releases reactive oxygen species. It has been shown to reduce angiogenesis and expression of vascular endothelial growth factor in some tissue cultures. Recent pharmacological evidence demonstrated that artemisinin derivative dihydroartemisinin targets human metastatic melanoma cells in vitro with induction of mitochondrial apoptosis. A pilot study on the use of artemisinin derivative artesunate yields promising result for the treatment of colorectal cancer. Module 5 Chemistry, Biogenesis and Pharmacological Activity of Diterpenoid. In this module, we will study in detail about Taxol. Taxol, synonym paclitaxel. Biological source, it is obtained from the bark of Pacific yew tree, that is Taxus brevifolia, belonging to the family Taxaceae. The chemical component 10-deacetyl-baccatin 3 isolated from the needles of Taxus baccata may be conveniently converted to Taxol via simple synthetic route. Now, chemistry of Taxol. Molecular formula C47H51NO14. Molecular mass 853.906 gram per mole. Taxol has following characteristic features, namely, first, it has taxone ring system. Second, it has a 4-membered octane ring. Third, an ester side chain at carbon number 13 of the taxane ring is a prime requirement for Taxol cytotoxic activity. Fourth, the presence of an accessible hydroxyl moiety at carbon number 2 of ester side chain renders an appreciable enhancement of cytotoxic activity. Now, biosynthesis of Taxol. Taxol is a diterpenoid which is synthesized via mevalonic acid pathway. Isopentyl pyrophosphate and dimethyl allyl pyrophosphate combines with each other and leads to the synthesis of geranyl diphosphate. By using Taxol synthetase enzyme, this geranyl diphosphate gets converted into Taxa 4,5,11,12 dinym, which undergoes several hydroxylation, acetylation and oxidation reaction and leads to the formation of intermediate, that is baccatin and 10-deacetyl-baccatin 3. 10-deacetyl-baccatin by the simple synthetic reaction leads to the synthesis of Taxol. Now, pharmacological activities of Taxol. Taxol is a primarily employed in the treatment and management of metastatic carcinoma of ovarian glands. It interferes with the normal breakdown of microtubules during cell division. It causes defect in mitotic spindle assembly, chromosome segregation and cell division. Paclitaxel solubilizes the microtubule polymer and protect it from disassembly. Thus, chromosomes are unable to achieve a metaphase spindle configuration. This blocks the progression of mitosis and prolonged activation of mitotic checkpoint trigger apoptosis or reversion of G phase of the cell cycle without cell division. It is also used in the treatment of breast cancer, usually after the observed failure of combination chemotherapy from metastatic disease. Paclitaxel is used as an anti-proliferative agent for the prevention of restenosis, that is recurrent narrowing of coronary and peripheral stents. A paclitaxel coating limits the growth of new intima, that is scar tissue within stents. Module 6 Chemistry, Biogenesis and Pharmacological Activity of Triterpenoids. Triterpenoids generally are obtained by biogenesis from 6 isoprene units. They are found to be share commonly the acyclic precursor ciculin, that is C30. The triterpenoids may be categorized into two major groups, tetracyclic triterpenoid and pentacyclic triterpenoid. Tetracyclic triterpenoids are the steroidal types with C27 carbon atom, while pentacyclic triterpenoids are triterpenoids with C13 carbon atom. Interestingly, both these types of compounds may easily combine with sugar moieties at carbon number 3 position to yield the corresponding glycosides. Nevertheless, the free triterpenoids are invariably associated with natural latex, resins or cuticle of plants. However, the steroidal types and triterpenoidal types may be distinguished by virtue of the fact that the former deals hydrocarbon on dehydrogenation with selenium at 360 degrees centigrade, while the latter gives either naphthalene or pinene as end product. Now, we will study in detail about chemical property and pharmacological activity of medicinally important triterpene, that is azadirachtin. Azadirachtin, biological source, it is a tetra-non-terpenoidal obtained from the seed of neem tree, that is Azadirachta indica and Melia azadirachtae, belonging to family Meliaceae. Chemical property of azadirachtin. Chemical formula C35H44O16. Molar mass 720.72 gram per mole. Azadirachtin, a chemical compound belonging to the limonite group. Azadirachtin has a complex molecular structure. It presents both secondary and tertiary hydroxyl group and a tetrahydrofuran ether in its molecular structure, alongside 16 stereogenic center, 7 of which are tetra substituted. Now, we will study biogenesis of azadirachtin. It is believed that the steroid tyrocalol is the precursor to the neem triterpenoid secondary metabolite, that is azadirachtin. Tyrocalol is formed from the 2 units of farnesyl diphosphate to form a C30 triterpene, but then loses 3-methyl group to become a C27 steroid molecule. Tyrocalol undergoes an allylic isomerization to form butyrospermol, which is then oxidized. The oxidized butyrospermalol subsequently rearranged via Wagner-Marvin 1,2-methyl shift to form apotriocalol. Apotriocalol becomes a tetra-norterpenoid when the 4 terminal carbon forms the side chain are cleaved off. The remaining carbon on the side chain cyclize to form a furan ring and the molecule is oxidized further to form azadirone and azadiride. The third ring is then opened and oxidized to form C-saccolimonides such as nimbin, nimbidenine and selanin, which has been esterified with a molecule of tiglic acid, which is derived from L-isoleucine. It is currently proposed that the target molecule is arrived at by biosynthetical conversion of azadirone into selanin, which is then heavily oxidized and cyclized to reach azadirachtin. Now, pharmacological activities of azadirachtin. It is a highly active feeding deterrent and growth regulator. It is used experimentally as insect control agent. It helps in the insect acidosis and growth inhibition. Summary. Terpenoids are the wide range of compound and possess variety of pharmacological effects in human being. Basic knowledge of terpenoids and its biosynthesis, chemistry and pharmacology has accelerated the pace at which the scientists have come to understand many plants' biochemical and metabolic process and their uses in the treatment of human beings. In this lecture, you have already studied in detail about chemistry, biogenesis and pharmacology of menthol, artemisinin, taxol and azadirachtin, which is used as a cooling agent, antimalarial, anti-cancer and growth regulator respectively. With all these information, here we comes to end today's lecture. Do keep in mind what we have discussed today. This is time for you for self-study. This is Anuj signing off. I will be back with one or more lecture in another series. If you want to learn more and enhance your knowledge, you may log on to our website, that is www.cc.nic.in, to download MCQ, quiz, LOR, videos and many more content. Till then, keep studying. Goodbye.